This is going to be a great session. I'm glad you all stayed and have lunch and so forth. For any of you who have heard an advertisement about probiotic food, or has taken antibiotics and noticed that your intestinal habits have changed, or you know that pathogenic organisms in and on our bodies are perfectly okay as long as they stay in balance with all of the other microbes that we carry, or you know something about the microbiome, and you think of it in human terms. We know that all humans, indeed all animals, have their own unique microbiome, and that can actually be used forensically, if you can believe that. Just as also do our houses and our offices. They have their own microbiome. And there are scientists working to understand how buildings develop their own unique microbiomes. And it's not surprising then for you to understand that plants also have microbiomes. By microbiome, I mean nothing more than all of those little small prokaryotic and eukaryotic things, the viruses, archaea, bacteria, fungi that we normally think of as microbes. Hence, microbe, and biome is the little world in which they live. Now, encouraging taxonomic diversity is fundamental to the philosophy of life. That's why we're all here, because we want to extend and encourage that kind of interaction between the various taxonomic components of our vineyards and our farms. And furthermore, the idea of homeostasis of these various taxonomic components, especially the microbial components, is really important to maintaining balance on our farms, and that the bacterial components of and the fungal components of our grapevines are really important to be balanced, and we want to make sure that when in our farming practices we don't disturb that balance, or we don't disrupt it too extremely. So, but what we are learning now is that the microbiome in wine grapes is really something to pay attention to. Scientists have looked at yeast and the number of yeast that you find in the vineyard and tried to relate that to what happens in the winery. But what we're learning, I think, is that it's more than just a few of the yeasts that contribute to the ferment. It's something much larger than that. The microbiome is probably a really critical component of terroir, and I'm trying to be kind of cryptic about some of these things because I don't want to give Dr. Gilbert's speech for him, but he will tell you about some of this kind of stuff. Now, why is he here? Well, it's because he is probably one of, if not the world's foremost experts in the microbiome. He is a microbial ecologist, I guess, I think that's the rubric that he goes by. And he has appointments at the University of Chicago, Argonne National Laboratory, the Woods Hole Biological Laboratory. He's a PhD, but he actually is a professor of surgery, I think, if that's right. So be careful, not all surgeons actually... He's one of the foremost thinkers and experimentalists in the microbiome. He's founded the Global Earth Microbiome Project. He's co-founded the Gut Microbiome Project, a hospital microbiome project, and he's a leader in developing technology for monitoring and understanding the microbiome in our built environment, the rooms that we live in, the houses we live in, the offices we occupy. Most important yet, Dr. Gilbert likes wine. And he's published on the contribution of the vineyard microbiome to the character of wine. And in fact, part of the way we're paying him for this talk is that we're giving him wine to take home. And so he's going to tell us about the vineyard microbiome. Dr. Gilbert.
Yeah, no, I don't come cheap as well, so I appreciate the contribution. I am English. Does everyone speak English here, or is it an American audience? Yeah, is one English, two English? Oh, yeah, that's good. So, yeah, no, it's my great pleasure to be here. I really enjoy speaking to growers, generally. I really enjoy actually speaking to the people on the front lines of whatever field that we're working in. I'm in a I'm a professor in the Department of Surgery, right? So I routinely speak with clinicians in that context, so people trying to make people better. But similarly, I work with people doing oil refinery aspects and hydrocarbon development, and I work with people looking after animals in aquariums or in zoos or actually out there in the real world. And so the work we do with growers and plant builders and farmers has been fundamental to developing that aspect of our research. Having to understand what it is that you want to know, right? Because I'm not a mind reader and I can't fully grasp what a grower goes through on a day-to-day or annual basis, is essential. If we're going to have a dialogue, we need an open dialogue, a two-way street where you talk to me and I talk to you, and we try, sometimes with difficulty, to understand each other, not just because of the complex accent I'm using, but because of the words we use, right? We need to be able to communicate with each other on a level playing field. And that sometimes gets lost. And I will fully vouch that it gets lost on our end a lot more than it does on your end. We've failed as a scientific community in many cases to communicate appropriately with the people we're trying to help most. And I will vouch for this, that patients in a hospital feel the brunt of that on a regular basis. And as you are all human beings, I assume, you will all at some point have probably felt that. And so imagine the frustration you feel when you don't fully understand a practice that's going on in a surgery or a therapy that you're having to undergo as a human. Imagine transcending that and putting that into practice, trying to look after your crops. There's fantastic parallels, and I think this is most important, fantastic parallels between how we treat people, for medical purposes, and how we treat plants. And in fact, if anybody on this planet over the last 200 years has grasped the appreciation of what bacteria and fungi can do for the health of another living organism, it's been farmers and growers. These are, you guys especially, were some of the first people to really embrace the need for a probiotic. Right? The addition of rhizobia into the soil in order to increase nitrogen fertilization, a natural approach to a solution to a problem, right? You had a problem, find a solution. We have a biological solution. I do a lot of work with chemical companies, and they are so sure of themselves that they can solve every problem with chemistry, they forget about biology. And then I have to come in and explain to them, look, dude, we've had like 4 billion years of biological evolution. You chemists have been at it for a couple of hundred. You really think you know all these answers? Okay, right. Then they show me the door. But in principle to that, we've started to really re-embrace our understanding of the role of biology in helping us to discern health, industry, growth and productivity in our farms. And so just yesterday I was giving a talk at the Milken Foundation about these very principles to a room full of growers. They were Californian growers. Anyone from California here? No? Oh, sorry, man. But I'm understanding there's a little bit of a division. Right. Yeah, exactly. You're at the back. Fast exit. Fast exit. But there were a lot of people like that in the room who are extremely interested in what they can do to ameliorate disease, to improve productivity, to transcend differences that are coming with climate change, and to enable them to be more ready with a changing market as well as a changing environment. Right? So this is something which is having a profound influence upon the industry. And I want to start by managing expectations. Right? I am not going to get up here and tell you we have a solution. We don't. This isn't, I've said this before, this is an open dialogue. We want to hear from you as much as I'm hoping you'll want to benefit and hear from the kind of research that the scientific community is trying to develop. We want to be able to understand fundamentally how microorganisms and plants exist, how they exist within the context of the kind of agricultural practices we see in place, and what that means for the market development, because we all need to earn money, as well as for improving our green footprint and changing and being resilient to the changes ahead in terms of climate. So we are trying to figure out a way to merge fundamental scientific inquiry, and I can get down to the level of an individual protein in an individual bacteria interacting with an individual plant cell, right? I want to take that and transcend it right back up to the scale of the growing system. So with that, I want to start by starting with humans. I want to give you some context because you are all humans, and I often find the best way to talk to people is to talk about them and their health, and then I'll get on to the nitty-gritty scientific bits, and you can fall asleep if you want after that. But when we first started to be able to sequence the human genome, right? Each one of you has around 3 billion nucleotides, 3 billion letters of your genetic code in every single one of your cells. It tells you how your body's going to grow. It tells your plants how they're going to grow. It gives the blueprint to their development, right? When we want to understand a link between, say, the genetic code and disease, we do an association study. And what that means is I'll take every single person in this room and I'll sequence your genome, and then I'll take your records of health. There'll be some people here who have suffered liver disease. There's some people here who may be predisposed to Alzheimer's in their family or Parkinson's. There's some people here who have a cancer diagnosis. And then we'll look for associations between the genes that make you different and those diseases. I do have a predisposition towards Alzheimer's, so does my mother. She gave me more than bad self-esteem. She's nearly 38 times more likely than the average person to develop Alzheimer's. Doesn't mean she's going to develop Alzheimer's, though. These are probability estimates. And we have to understand this. Your genome it's like the stage against which you'll play. Well, I'll get very philosophical. We heard about philosophy earlier, right? I can't remember who was saying that, but this is a play that your body is playing out, but it's not doing it in isolation. It's doing it in a continuum, an ecological continuum with the world, okay? And so, just because I have a predisposition and my mother has a predisposition towards Alzheimer's doesn't necessarily mean that we're going to get it. Although my sister would tell her that she is going mad on a regular basis, but then I never thought they got on very well, so who knows. Now, the complicated thing is, when we try and look for the reason why some people do develop this disease and some people don't with exactly the same genetic profile, even identical twins can suffer this fate. One can become sick and the other one doesn't become sick with the exact same genetic profile. We have to take into consideration that you're not just a human. You heard this earlier. You're about 100 trillion bacteria also inside your body. They do outnumber your human cells, and the estimates range by about 2-fold to 10-fold, and we're still trying to figure that out. It's very hard to count the number of bacteria in a human being. They're very small, they're very, very, very small, and we find it very difficult to make those calculations. But what's interesting about the bacteria, 100 trillion cells versus your 10 to 20 trillion cells, okay? Those 100 trillion cells, each of them has its own genome, right? That tells it how to grow, tells it what to do. And in fact, the microbiome has 100 times the amount of genetic information that your own genome does. And as we heard, each one of you has a unique microbiome, okay? So that unique microbiome is interacting with your genome, and it's changing how your body develops from pretty much the moment you're born. And I'll talk about that a bit in a minute. But that association is incredibly important. And by doing what we would call microbiome-wide association studies, what I mean by that is, like this genome-wide association study I talked about, take all your genomes, relate the changes, the differences between you two diseases. Like that, people in the world, like that, we do a microbiome-wide association study. We look at each of your microbiomes and we compare it to your diseases, right? And then we link that to your genes. Another layer of complexity is a 3-fold comparison. And when we do that, we can start to actually see why certain people have a genetic predisposition but don't get the disease, because their microbiome differentiates them from the people with the genetic disposition that do get the disease. And this is fundamentally changing our perspective on medicine. I actually started out my career as a butterfly specialist. Right? Seems weird. I was working on butterflies in Africa, the ecological divisions, why certain butterflies were found in certain places. And then I became a marine microbial ecologist. I started to look at bacteria in the ocean. And now I'm a professor of surgery. Right? Why the hell would a surgery department hire an environmental ecologist? Right? Somebody who looks at why organisms interact with other organisms and their environment. Well, because we are an ecosystem. Right? And trying to understand why your disease occurs, trying to understand how your life shapes what diseases you're going to be susceptible to has become fundamental to medical practice. And genome-wide association studies combined with microbiome-wide association studies have led to an identification of the microbiome being a component of all of these conditions, being affected by them, being actually causing them in some cases, and leading to the development of conditions which seem almost distant and impossible to touch. Even depression and anxiety, even the way you believe you think you feel. I'll talk about that in a second. I have a nice nugget. Some of the most exciting things that we've been uncovering, for example, are the role that the microbiome can play in cancer. We know, for example, that if you have certain types of bacteria in your intestine, in your colon, they produce chemicals that can lead to the development of precancerous polyps. Okay? And when we see people with those bacteria in them, if we knock them out, they no longer get precancerous polyps, and their likelihood of developing colorectal cancer drops through the floor. It becomes almost negligible. We also know that in treating things like melanoma, where we can use your own body's own immune system by producing antibodies to the actual cancer and pumping them into people, we can actually train the immune system to attack the cancer itself and wipe it out. But some people don't respond very well to that therapy. So what we've actually identified are bacteria that we can add into a patient that stimulate the immune system, cause, if you will, an allergic reaction, but magnify that immune system response so that that antibody and the bacteria, the probiotic, can stimulate the immune system to kill off the cancer. And with that, we get almost a 95% cure rate. And this is transcending. I don't say it's the beginning of the end, but it's definitely the end of the beginning for the fight for cancer. We're now starting the main war. And everyone says this, in 10 years we'll be done. But we are starting to develop novel combinatorial therapeutics which are going to have massive implications. Depression and anxiety, a major problem in our community, for many, many different reasons. There's an old adage that the way to a person's stomach is, the way to a person's heart is through their stomach, or whatever it is. The way to a person's brain is through their stomach. Well, it's true. We can change the microbiome in an individual person or animal's stomach, in their intestine, and it will alter their behavior, their mood, their anxiety, their depression. I can actually make a mouse stop feeling depressed. I know it's weird, a depressed mouse. By altering the bacteria. I can actually change mate preference for a fruit fly. We heard about fruit flies earlier. You want one fruit fly to fancy another fruit fly? I can make that happen. I can set that up like fruit fly Cupid. We can add bacteria into a fruit fly and change its mental behavior, its behavioral activity, and change who it prefers. Maybe this happens in humans, we just haven't been able to uncover it yet. So with depression, we've actually uncovered a significant number of microbes which lead to depression in individuals. In people that are either genetically predisposed to the condition, they have these bacteria in them. These bacteria produce certain chemical reactions which lead to almost a neurotoxic effect, causing a significant suppression of their ability to maintain a mental stability. We're leading this into clinic right now, trying to add these bacteria into patients as a probiotic to see if that will alleviate those conditions. In our preliminary investigations with postpartum depression in women, obviously you can't have postpartum depression in a man, We haven't got that far, that'd be weird. But after pregnancy, we have a significant problem with depression. We've actually been able to alleviate depression in our small cohort studies by almost 70% with the addition of this particular probiotic. So it has a real ramifications. Surgical infections, still there are tens of thousands of problems with people acquiring a pathogen during their stay in a hospital while they're undergoing surgery. The very place they should not be getting sick. And they go in there and they come down with a multi-drug resistant pathogen and it can kill them, right? So we've started to uncover ways in which during surgery, by augmenting the microbiome, by changing that microbial ecology inside the patient, we can prevent pathogens from taking hold. We can prevent the person from developing a sickness. Why? Because about 90% of surgical infections originate in the person's body. This is a bacterium they had with them when they came into surgery. And if you think the surgery is stressful for the human, it's incredibly stressful for the bacteria as well. So, put your heart out to those organisms that are suffering horribly on the operating table, 100 trillion of them. But when we figured out a way to keep them happy, right? We keep them fed. We add in nutrients into the colon, into the lower intestine, where the vast majority of them are. And when we do that, they stay in a state of happiness, a state of bliss that prevents them from attacking the host. We keep them fed so they don't feel starved. When they feel starved for nutrients, they attack the host to get the nutrients back. Like a riot in an inner city environment for deprived people, these organisms will fight to get what they can back from the host. And in doing so, accidentally probably, they kill it. And doing this, we've actually managed to alleviate surgical infections in our preliminary cohort studies by 90%. So those 90% of cases where this originated from, we have a nearly 100% record of effectiveness for this treatment. It's going to be a long time before we can get these things into the clinic, but we're working very hard to try and improve the rapidity, the speed at which we move this out of our small studies and into regular practice. But there's hope. And the hope comes from looking at the microbiome ecology. So where does the microbiome come from? You're all born sterile, for the most part. And the bacteria that you first interact with are the ones from your mother, right? The method of your delivery into this world actually affects what bacteria you're colonized by. If you go via the standard route, you're colonized by your mum's vaginal microbiota. If you go via caesarean section, you're colonized by bacteria from her skin. And that stays with you for a few months. That's your sourdough bread, that's your inoculant, right, for your wine. Be a bit of a weird inoculant, maybe I shouldn't make that analogy. Right, distract that. But if we can figure out a way in which that affects the development of the child, which we are doing, then we'll have massive implications. But it's not just that, it's what environment you mess around with. That dog that licks your face when you're 2 years old and won't stop bugging you. These mud pies you make outside in the soil, rummaging around. That interaction you have with the environment fundamentally affects what kinds of organisms will colonize and lead to the development of your microbiome. But over the last 100 years, we've been doing more negative processes. We've been isolating ourselves from that world. I'm actually preaching to the wrong audience in this case, because the vast majority of you, I reckon, are very integrated into the soil and the environments in which you work. But the vast majority of people spend 90 to 95% of their lives indoors, in this kind of environment, with sterile air being pumped in. Not sterile, but very dry, very clean. And we're missing that exposure. Our children are missing that exposure. We know that children that grow up on a farm actually have a 50% reduction in the likelihood of developing food allergies and asthma. And in fact, on this particular Amish farm in northern Indiana, we see that their population has under 5% food allergies and well under 1% asthma in their population. The average for the US is about 7% for asthma, right? In this Amish population, it's less than 1%. For some reason, these children are not developing the conditions they're genetically predisposed to develop. In fact, for whatever reason, we think the exposure on this farm is fundamentally altering the development of their immune system, allowing them to respond appropriately to the stimuli which would normally cause them to overreact. I actually take my kids out to this farm. It's about 2 hours east of Chicago, and rub their faces in the dirt as much as possible. Because otherwise they won't get off their iPads, you know, it's bloody awful. But these, you see children like looking after the animals. I mean, they play an active role in the management and the development of this farm. They have to, there's nothing else to do. So they work pretty much constantly. There's an interesting thing, the Hutterites up in North Dakota live a very different life. They have the same kind of removal of mechanical objectivity from their lives, post-plough. They like the plough, that's good mechanics, but everything after that didn't work. But only when they're at home. When they come onto the main farm, they actually have a very mechanized environment. But only children, because it's mechanized, only boys over the age of 14 are allowed on the farm. So little boys and little girls never get that farm exposure. And the Hutterites, especially in this community in northern North Dakota, despite the fact they come from nearly the exact same genetic lineage as the Amish in northern Indiana, right? The Hutterites in North Dakota have 37% asthma in their population. And this is fundamental. These kids are being comprehensively isolated from the environment, which as a lineage they're expecting to see. Their ancestors grew up on farms. It doesn't take much of a thing to say, well, they're adapted to being on a farm, okay? And many of us, still, in America, in Europe, we come from these agricultural lineages, right? And when we remove ourselves too far from what our bodies are expecting to see, we get this overreaction. It's not rocket science. Well, it's much more complicated than that, it's microbiology. But for us, it's common sense. And it's exactly what my mother brought me up to understand. Go outside, stop watching the TV, and start mucking around in the dirt, okay? And if you practice that enough, you'll find there's benefits. And I'm sure of that. We actually have an article coming out in the New England Journal of Medicine in a few months, or a few weeks, that talks about this study and actually proposes this as a strategic initiative for enabling children to benefit from this kind of life. So what does your built environment world look like? Well, the world you're in right now looks like you, unfortunately. It looks like your skin. Each one of you, right now, are emitting in a cloud around you around 38 million bacterial cells an hour. So in the time you spent sitting in this room today, you've actually significantly increased your microbial similarity. All right, you can stop rubbing each other, it won't help very much. You're sharing that microbial world, right? The air around you right now is full of bacteria. The surfaces are full of bacteria. You think you can sterilize it? You cannot. It doesn't work, right? All it does is leads to an enrichment for the microbes that are in your mouth, on the skin, and on the skin around you. The answer, in our impression, is to take this sterile world and try and improve the microbial diversity in it. That's a very controversial thing to do. Spent 100 years trying to go in the opposite direction. Now we don't know yet. We have no roadmap to come back into a more biodiverse experience. But if I can't get all the kids in South Chicago to go to the farm, maybe I should be bringing the farm to South Chicago. And this led us to an appreciation of our need to really understand what that microbial world actually looks like. And so 6 years ago, when I came over to the States, I was working in industry beforehand, and the University of Chicago offered me a position to come over and do something I wasn't used to in industry. So, hey, just come over and think. What's the biggest problem you can possibly think of? Go out and do it. And I chose what is probably considered one of the most impossible problems, but what the hell, they gave me a task. We said we're going to figure out what the microbial world actually looks like. We're going to catalogue it. All right, we're going to produce a Noah's Ark, a Darwin's Guide to the World. Let's just find out how many bacteria are out there, which ones, where are they found, and then we want to use that to change the world. No small dreams, right? Let's just go big or go home. And so the Earth Microbiome Project came out of a brainstorming session where we wanted to be able to understand the microbial diversity of this planet. We have over 400 collaborators now. We sampled over 50,000 types of environment. We have over 6 billion sequences generated. This is little nucleotide sections of the genomes of these organisms that we've sequenced. And we found approximately 27 million species. And in fact, a recent paper that came out in the proceedings for the National Academy took our data and extrapolated it with some very complicated math. I was the reviewer and I'm still struggling with it, but took that data, extrapolated it and said, if this data is true for this number of sites, there are approximately 1 trillion bacterial species on this planet. Which, if you think about it, means that since the earliest days of our planet, there's been a new bacterial species, or about 3 or 4 new bacterial species cropping up every single day, which is impossible. But if you look at it from an exponential perspective, these microbes are constantly diversifying. Right? So in every environment on the planet, they are growing and they're developing new species, including in this environment. So if you think they're just skin bacteria going to stay just skin bacteria, it's not. Right? The microbes are evolving to survive in this ecosystem. And any ecosystem you've built, any ecosystem you've created, they're evolving to fill that niche. So we've used this kind of information to start to help us to understand the interaction between agricultural land and the microbiome. And from this perspective, in work we published a couple of years ago in Science, we started to try and relate that to changes in climate. We thought, if we can understand what microbes are there, can we link them to actionable evidence about what we could do with the world? So we collected samples from relic tallgrass prairie sites around America and the Midwest. And this meant creeping around old Civil War graveyards and railways with a soil core and being a little bit weird. But what we found out was that the vast majority of the microbes that really would have correlated with the tallgrass prairie regions in about 1840, so we're time traveling here, would have been the Microbiome. And these are interesting, they're very hard to culture, right? They appear to flourish where you have a high carbon and low nitrogen ratio. They're very good at carbon metabolism, they're very bad when too many nitrogen compounds come in there. But without them, the tallgrass prairie plants don't grow very well. They're not particularly resilient to frost, they're not particularly resilient to drought. It changes how those plants respond to climate, okay? And we found that by linking the microbiome below ground to the climate across this region, we could predict exactly where these certain types of plants would grow. And by using this, we started to help inform organizations that want to restore prairie sites and explain to them, hey, if we just package up these spores of Microbiome and plant them in when you're replanting the prairie land, it can have a significant effect in improving the productivity and the resilience of those plants. And what we found is that by adding these organisms back into certain prairie sites, we've actually been able to even reduce the burden of invasive species, because the prairie plants are now more competitive in that environment. And I'll talk a little bit about what we've done in crops for that towards the end. We've also done this just recently in China. The scientific community knows no boundaries. I'm an immigrant into America. We work very closely with the Chinese on trying to develop research across these boundaries, which can actually be impactful at a global scale. So here we were looking at microbes across forest sites, 110 forest sites from the northern part of China to the southern part of China. And we identified lots of different microbial species across that gradient and used that information to build a map. And this seems very complicated, you don't want to understand it, I hardly understand it. But it's a network map that it helps us to understand how all of these microbes interact with each other, and how these are fungi, these are bacteria and archaea, which are like bacteria but very different. It's very complicated. I always had trouble understanding that. So if you look at archaea and bacteria under the microscope, they appear identical, right? They even have the same kind of cellular apparatus inside their cells. But they are more different to each other than you are from a bacterium. It boggles the mind. But when we look at these kinds of things, we find that there are a huge number of interactions between the microbes in the soil, the microbes in the plants. And we've linked these interactions to climate. So what we're seeing here is a gradient from a high degree of connectivity between the plants and the microbes to a low degree of connectivity. And what we found was that this was heavily linked to changes in rainfall. So when you go down in the south area of the country, where you have a lot of rainfall, the plants and the microbes are less connected. And it might seem like that shouldn't be. When you go up into the northern part, where it's much drier, the plants and the microbes are much more connected. There's much more synergy between them. Why? We hypothesize, and we're still working this out, we hypothesize it's because when there's less moisture around, the bacteria and the plants have to cling to each other to actually have any relationship, okay? Because there's no water to carry the nutrients between the plant and the bacteria and the plant. But when the plant's embedded in water, then the plant can accept nutrients, and microbes can accept nutrients at a much greater distance. The water acts as a medium that carries the nutrients between the plants and bacteria, enabling them to become much less reliant on one particular co-association. So it's like bamboo and pandas, right? The bamboo and the pandas are very reliant upon each other. Okay, without one, the other can struggle. Well, maybe the bamboo would be alright, but the panda would be fucked, quite But if we look at that, right, that's like in the north, where they're really reliant upon each other. The plants need the bacteria need the plants, because without that co-association, neither of them can grow. But when they're down south, the reliance starts to break down because there's much more food at the party, right? The plants can rely on many different types of species, and those co-associations start to break down. This is surprisingly simple finding. This surprisingly simple finding is actually helping us to understand how these ecosystems are going to change with shifting climate patterns. Understanding how particular plant species are going to manifest their differences when you get much more rainfall and your land becomes more waterlogged for longer periods of time. I don't mean anoxic, I don't mean it's going to become saturated to the point of anoxia and produce methane, but it will, when the land becomes more waterlogged, you'll see this significant differentiation. We've done this across North America and Tibet, and we came up with some fun little vignettes. So this is Tibet from 1900, in the years up there. Average yearly changes in the microbial communities and plant dynamics across this system for 100 years, 111 years. It's a pretty picture, right? But actually what it tells us is that the network relationship between the plants and the microbes has changed substantially over this time frame. I'll rerun that. So very early on it was very stable, as the climate was stable. When the climate started to change and agricultural activity in this environment started to change, this environment started to undergo massive dynamic fluctuations. It's currently in what we would call a dynamic disequilibrium, right? The ecosystem on the Tibetan plateau is struggling to maintain any stability. It's fluxing, it's dynamically fluxing between one stable state and another. And that makes it much harder for us to be able to predict what the hell's going to happen, because the system doesn't have any predictability to it. When it's in chaos like this, it's difficult to understand what the next growing season for some of these farmers, or the next herbivory season for the cattle ranchers is going to be like, right? And it makes the economic stability of that region much more difficult to predict. But using these algorithms, we can start to at least put bounds on those predictions, linking changes in productivity across this region to climate. We've done this for North America, and we can actually forecast into the future what the system's going to look like. So you guys are over here somewhere, right? So this is the current richness of microbes across continental US, right? For you over there, it's that one on the left. So these are the number of species you find. You don't find many in the far Arctic, it's too bloody cold. You find lots down when you get to Mexico, nice and warm and lots of moisture in those hot regions. And this, on the right-hand side, is the change in that microbial richness. So the red areas are where you're going to get a lot more species turning up, and the blue areas are where those species are going to disappear. So in regions around the equator, you're going to get more species turning up, but it's pretty much going to be business as usual overall. But in regions where you have desertification, the number of species are going to go down due to drought. And in regions up here in the north climate, we're going to get a significant increase in the number of species and co-associations with plants as that system starts to melt. So these are helping us to predict across large scales what might happen. I'll leave that. So I'm here to talk about wine, right? I forgot about that. Wine. I didn't have any with lunch. So when we first started doing this, back in about 2011, I was just looking for excuses to go out to wineries. It really wasn't a scientific justification for a study, but it turned into something much bigger than that. We were looking for sites to do agricultural activity for the Earth Microbiome project, and one of our colleagues worked with the Norfolk winery people in New York, in Long Island, and they were very accommodating in helping us to come out there. Gilles Martin, the lead winemaker, was incredibly gracious, and they allowed us to come out and start sampling their plants on a regular basis, and we built up a huge database of information, I think the largest to date, about the kinds of bacteria we find on the grapes, on the leaves, on the roots, in the soil. And this helped us to start to ask questions. It was a baseline. It started to help us to ask questions about how that climate, how that environment might respond. We're still working on those. What was interesting though is I hear a lot about the microbes on the grape. Why? Of course, because it affects fermentation and that's the end product. But I hear very little about the microbes actually in the soil and in the root system. And why is that important? Well, we just talked a lot about how microbes affect children, affect people, affect health. This is just another person, right? I don't know how many of you have very personal relationships with your vines. You can get very lonely out there. But you do have that relationship, right? And these things have to be taken care of. They're very sensitive, and we need to be able to provide the right environment. And the right environment to do that is probably in the soil, the terroir of that system. The right nutrients, the right microbiology, you have the right product. We started to look at this across these different environments. All of these samples are above ground samples, so the flowers, grapes and leaves. All these are below ground, these kind of species we find, don't worry about that. This is the association with the plant system. These are all the bulk soil and the root zone soil right there in the middle. And the little dots around the outside are all different bacterial species. And so what is that? Is that a fly? Sorry. No way. The bacteria are very highly co-associated with the root and the soil system. And the roots are picking up the bacteria from the soil and allowing them to colonize inside the root, leading to a very significant soil-like microbiota in the root. And that's all the blue samples here. But then as we get up closer to the grapes and the leaves and the flowers, the microbiome significantly shifts. But what was interesting from this study was that still all of the bacteria we found up there, or 97 to 98% of the OTUs we found, originated in the soil. In the soil, they were very rare, but they were there. And then they were being appropriated from the soil and spreading throughout the plant and colonizing the different parts of the plant. Now, if I take your mouth and your stool, they're as different from each other as a coral reef microbiome is from a Nebraskan prairie microbiome. Okay, they're very, very different. Same with a plant. If I look at the berries and I look at the roots, the microbiome is incredibly different. But the origin is still the same. Just like with your baby coming into the world sterile and being colonized by mother and the environment in which it finds itself, this plant is being colonized, and that's going to affect how it develops, how it grows, what its resistance to stress is, what its resistance to disease is, what its productivity is. And we need to understand that. We looked at this across many different vineyards, trying to control for things like the soil type and the amount of carbon in the soil, trying to control for varietal and even vineyard in of itself. Very, very complex to do. We were just discussing this earlier. It's incredibly hard to go out into the environment unless you're doing it at a massive scale and control for all the variables which could be influencing the microbiome. Just as it's very difficult for me to go out into this population of people and control for all the variables which could have affected your microbiome and could be leading to disease. So these studies aren't necessarily ideal, but they help us to provide a baseline. For example, we found that pH was a strong influencer of microbes found at this particular vineyard. And if you go into that soil, it does have a slightly lower pH, and that was significantly shaping the bacteria and significantly shaping the bacteria that colonized the vineyard, the vines. Right? And we actually saw very different bacteria and different fungi associated with the berries in that vineyard because the soil system was different. Right? But it was the microbes, we think, that are playing a more significant role because nutritionally, that soil was identical to all the other soil types. It was only the pH which was different. When some colleagues of mine who worked with me on that study at UC Davis and their enology centre did this looking just at the must, right? And looking at the bacteria and the fungi across different regions. And in this they do show that region matters. That the Bacteroidetes, the Firmicutes, these organisms, these bacterial communities which are found in different spaces are associated with the different regions. So the coastal area, the Sonoma area down here in purple, the Napa area up here in blue. The environment was selecting for different types of microbial communities which were colonizing the plant and then being associated with the must. This changed the paradigm of that environment and fundamentally altered the types of microbes we find in it. And the grape variety also had an effect. We had to understand that. So Chardonnay, Cabernet, Zinfandel, in this particular instance, affected the types of microbes which could colonize those plants. This is important. Identical twins, monozygotically identical twins from the same egg, identical twins share more microbial similarity than non-identical twins, or just siblings. Okay? This suggests to us that genetics does play certainly plays a role in shaping what bacteria can colonize the body, just the same as it does with these varietals. The genetics of the plant plays a factor. We have to understand every aspect of it. Getting each one of these varietals genetically sequenced and then having their microbiome sequenced would be exactly akin to what we're trying to do in medicine. The National Institutes of Health, we're just running a precision medicine initiative, sequencing the genome and the microbiome of over 1 million people. And doing this is going to fundamentally change medicine, while doing this is going to fundamentally change agriculture. Right? We just need to be brave and take that leap of faith. If you go into the winery, after the grapes have been picked, you can start to do analysis. So this is the Robert Mondavi Institute for Food and Wine Science down at UC Davis. It's a big, fancy place. What they did in this study was they took the environment inside the winery and tracked what kinds of fungi and what kinds of bacteria are found in different sites. This is the built environment, right? Somebody built this ecosystem and wanted to see what was happening. Again, work from David Mills at UC Davis. So in this, they tracked from pre-harvest to harvest to post-harvest the kinds of microorganisms and their distribution across that environment. If I zoom in on that, these are bacteria on the left, these are fungi on the right. You can't really see the names of these organisms, but you can see that things do change pre-harvest to harvest to post-harvest, as you'd expect. But the environment's never sterile. There's always a microbiome there, and many of those organisms play a significant role in the fermentative process, right? And we've known this, right? I work in cheese in and around Naples in Italy, cheese microbiome research. Again, because I wanted to go to Naples, Italy and work on cheese. Can you blame me? So we did that for a very particular reason. We started to look at how the microorganisms in some of these very ancient cheese environments, cheese fermenting environments, what role they were playing in the actual process that those cheeses were undergoing. And it was fascinating, right? The cheeses were being inoculated by microbes that were actually present in the environment, not present in the original Curzon way. And this same process is happening here. We find that when the wines are actually developing, when you get the fermentate, they're actually being colonized by microbes not just from the plant, but also from the winery. And that could be changing the fermentative process. The subtlety of that change still has to be determined, because unless you're, is anyone here like a world-class somm? Do we have any? Not everyone's like, no, I just like drinking wine, right? It's real hard to tell the varietal differentiation sometimes, okay? Especially in certain very subtle blends. But people can do it, but it's still a subjective characteristic. We're trying to find, as scientists, objective characteristics which we can map. A lot of that's down to the chemistry of that fermentated product, trying to link that to the organoleptic properties that we'd associate with, the flavour, the sensation, the je ne sais quoi, whatever. That's about the limit of my French, I apologise. But trying to find those links is important, but we haven't done it yet. So trying to understand that link between these subtle variations in inoculant, these subtle variations in fermentating characteristics in organisms, and what it actually does to the wine still needs to be done. And I'd argue that's the frontier that we need to be working on in this space. We find things like Cerevisiae and Coccus, these fermenting organisms, we find them very obviously significantly more abundant in fermentators, hoses, filters, pumps, areas. Tend to be similar to the kind of equipment we use to crush the grapes, right? So these are organisms coming in from those grapes. I find this, my mother, usually when we publish an article, I bring up my mother a lot, I probably have problems. My mother would publish an article on the human microbiome and goes, "We knew that, why did you spend $5 million trying to find that out? I could have told you that 30 years ago. Go out and get a dog, the dog bacteria will help you, it's fine." Yeah, well, so you didn't know it was bacteria, Mum, you just like the dogs. Now we can show that dog bacteria effects improve the ability of children to prevent asthma. Well, in this instance, we know that we should expect to see some of these fermentative organisms there, but how they actually colonize the fermentate and what role we can actually play in manipulating this event and actually using it to mechanistically alter the process is still unknown. And so actually having this information is incredibly valuable. Okay, so I'm going to finish up because I've been speaking for a while, but I just want to point out a couple of examples that we haven't done in wine yet, but these are studies which need to be in wine and point out some very interesting characteristics. Working in Arabidopsis, which is like our lab rat for plants, right? We've basically shown that under drought we can have bacteria that can alter the tolerance of that plant to drought. If we add these bacteria into germ-free, these are Arabidopsis plants that don't have any bacteria, any fungi associated with them, in sterile soil. If we add these bacteria to the plant resists drought significantly better than a plant without it. It keeps on growing. That bacteria stimulates the plant's immune system, right? And it alters the plant's growth characteristics under stress, actually enabling that plant to survive and propagate and become far more productive under short-term drought conditions. Understanding and being able to organize that characteristic is going to be fundamental to changing how we practice this process. Fungi can improve salt tolerance. Obviously not usually very relevant for some of the vineyard environments we work in, but there are some of the vineyards that get significant salt spray on the North Fork in Long Island. So we were working with them on trying to understand our arbuscular mycorrhizal fungi and the symbiotic relationship it forms with the plant and its effect upon the plant's ability to cope with elevated levels of temporarily elevated levels of salt in the soil. And this is, again, a process we can mechanistically devise in order to affect the productivity of that plant system. We've been working a lot also with smallholder tropical agricultural groups in Africa and Southeast Asia, trying to understand how fertilization strategies affect the biodiversity of the soil and how that concomitantly influences productivity. Obviously, some of these people, this is their entire subsistence, and so being able to optimize the strategies for fertilization, which obviously costs money, to productivity through the route in which is most important, which is I'm not just feeding the plants, I'm feeding the microbiome in the soil and the microbes on the plant as well. Being able to understand that link has become fundamental. So being able to model this relationship between the amount of nitrogen added, for example, and the diversity impacts upon the bacteria and fungi, and be able to use that to generate predictive statements that can actually be used in agricultural settings has become a priority. I just want to, I have a small, what's it called? Smoking problem? No, I don't know. The University of Chicago would kill me. I also work for the federal government and that would not go well. So I have a small conflict of interest and I'm an advisor for this company. But I just wanted to point it out because I became an advisor for this company because I started to help them to characterize certain organisms they produced. And the principal of this company, Grosentia, is a professor down at Colorado State University who identified a couple of organisms and then brought them to my lab to have them characterized. And we worked on what these organisms actually did. These organisms, what they did is when they were put in the soil, they found, tracked down, and activated phosphate sources in the soil which were usually recalcitrant. Usually no plant and no other organisms could actually get at these phosphate sources. There's a lot of recalcitrant nutrients locked up in soil. By adding in the right kinds of microorganisms, you can find ways to unlock them, but often we don't know what they are. So we sequence the genomes of these bacteria, and then we combine them in very specific combinations so that they would support each other. And when you sprinkle this on the plant, it grew far more vigorously than a plant without this solution on it, or a plant with just any old bacterial community on it. So after we produced that, he went away and in secret formed a company, and then told me later, which is nice. Brought me on as an advisor and not as a principal. This company is now making, I think, $80,000 a month, and it's only been going for about 2 years. But in 18 months after the inception, they produced this product, Mammoth P. Because he's from Colorado, he found a niche market. Right? They now have a competition each week to increase bud yield. So people who send in the biggest buds on their Facebook page get a free week supply of Mammoth P. Okay? The only reason he did this is because it's very hard to break into the big agricultural market with a small product. Right? You've got to wait until someone shows interest, they buy you up, and then Monsanto would take this and go through the regulatory processes to enable it to be effective. And so he focused on this. But through some of my wine connections, he's started to look into winery development. And so we've been working with a couple of the Californian wine groups and trying to see if there is a role for this, to be repackaged. Not quite so, hippie, but something that's a bit more sophisticated, maybe. But the idea is that by utilising this on poorer soil environments where the phosphate concentrations are more recalcitrant, you can utilise this to significantly increase yield in different field sites. So sites which are growable, and I fully agree with turning, with the speaker was up here on turning some of your land back into wetlands. If it's not an effective growing environment, don't use it. That is a brilliant strategy. But there are sites which are still effective growing areas which could be brought back into production, and this is one way we're trying to do it. So I wanted to bring that up. I do have a conflict of interest. I don't suggest you go out and buy it, but we are exploring commercially the commercialization of this science in order to rapidly advance application. So, last slide. Managing expectations, okay? I'm not here to solve your problems. I'm here to listen. I'm happy to listen to what the problems are. I can't solve your IBD, if you have IBD, but I'd be happy to talk to you about what the research shows. But I can't work alone. I need to actually work actively with the growers in the field. When I put a proposal into the National Science Foundation to do some of this research, we didn't have any links with any growers. I had to go out and build those links and establish that trust, okay? But it is a two-way street. If you have questions, people are out there, researchers are out there, young, established researchers, young up-and-coming ones, that want to work on interesting projects. And I get routinely asked on a regular basis very interesting questions that I think there must be a solution to. For having that dialogue and opening that dialogue, and maybe this community could be an excellent avenue for focusing that dialogue into appropriate locations. And we need to be brave, we need to make significant investments. That's very easy to say, very hard to do. But even if you make an investment, as any venture capitalist will tell you, doesn't mean it's going to work. Right? You've got to hedge your bets and spread your investment around a bit and try different scenarios. And if there's anybody in the world that understands that better than anyone else, it's farmers, right? Hedging your bets is a key strategy to maintaining productivity and economic stability. So having and embracing that relationship with the world, I think, is incredibly important. Okay, I will leave it there. Thank you.